Welcome to the exit seminar for John Ertle. He's defending today for his master degree in horticulture science. Let me introduce John before he starts the presentation. John Ertle is from Cleveland, Ohio, and then he was graduated from University of Akron in December 2017. Bachelor degree from biology as a major. And then during his undergraduate time in University of Akron, he had some research experiences in biochemistry lab, as well as ecology lab. Didn't have a chance to learn the exciting area of horticulture crop science until he had a working experience in a startup company in downtown Akron, Ohio, where he was involved in hydroponic lettuce, basil, microgreens, and vertical growing farming using NFT systems. And then he was very interested in that advanced technology of growing crops under controlled environment. And then he started looking for a graduate program and then discovered that a new program in Ohio State, which is my program, and then joined in August 2018, year 2018, as a master's student. And during that program for master degree in Ohio State, he participated several conferences, including NCERA 101, North American Committee for Controlled Environment Technology and Use, 2019, and then also International Symposium on Vegetable Grafting held in North Carolina, also 2019. And then we'll be presenting a few more papers in American Society for Horticulture Science this year. All these presentations out of the work he did for master degree. So you will be learning what he did for the past 2 years on the vegetable grafting technologies. So his presentation or thesis title today is the effects of short-term chilling stress on seedling quality and post-transplanting growth and post-transplanting growth of grafted and non-grafted watermelon. So if you are ready, John, it's all yours.
Awesome. All right. Well, thank you so much for the introduction, and thanks to everyone who is taking time out of their schedules to come and listen to me present today. I'm very excited to present my research on watermelon and grafting over the last 2 years. And so first and foremost, if you want to go ahead and pin my speaker window up here, that's just going to make sure you don't cover any content throughout the duration of this presentation. And as for the content itself, I'll be starting with an introduction and a background on why we're doing this kind of project. And then I'll go through 3 distinct experiments. The first being the use of 3 degrees Celsius chilling, and then the second experiment with 1 degree Celsius chilling, and then a final third experiment using mixed chilling temperatures and durations of chilling. And then I'll go through a conclusion section as well. So just to introduce grafting a little bit more, grafting is a unique agro-technology that is rather interesting, but basically you take a plant of interest or a scion that's going to produce some kind of vegetable crop for you. In this case, this is a watermelon scion. But you want to take advantage of the better root system of a rootstock plant, which in this case is interspecific hybrid squash. So by making incisions on the scion to remove the root system and removing the growing shoot tip of the rootstock, you're able to actually combine these plants and heal them inside a controlled environment to form a grafting union, at which point the plants essentially merge into one, and you're able to treat them as a normal seedling for your growth and development. Now, one of the many reasons you might graft a plant is to reduce soil-borne disease incidence, which is one of the most common reasons that we graft today. So in these top 2 photos, you can see watermelon roots that are highly affected by root knot nematode present in an open field soil setting. But in the bottom here, these are plants that are grafted onto a rootstock resistant to root knot nematode. And so in this case, there's no gall formation and the roots are growing healthfully and are able to allow the plant to continue growing normally, even though that there's disease pressure in the field. On the right here, this is a field infested with Fusarium oxysporum, which causes Fusarium wilt. On the left, you can see grafted plants that are grafted onto a rootstock resistant to Fusarium wilt. And on the right are non-grafted plants that are susceptible to Fusarium. In this case, the grafted plants are clearly having much more regular vegetative growth and are going to produce the fruits as you'd want in the field setting, while the plants on the right are suffering heavily from the disease pressure. Another reason you might graft a plant is to increase yield. So on the left here, we have two different tomato plants, one grafted and one non-grafted, with the grafted plant having a much higher fruit load and yield than the non-grafted plant on the right. And actually, this is one of the main reasons that grafting has been adopted in a lot of open field settings. So on the y-axis here, we have yield in kilograms per meter squared of watermelon in an open field in Arizona. And on the x-axis is the harvest date. So this is cumulative yield over the course of a season. And we have two different non-grafted options here, in which case the maximum yield throughout that season is about 3.3 kilograms per meter squared in the same field, while the grafted varieties are producing 6.5 or 7.9 kilograms per meter squared in the same field in the same year. This accounts for a 97% or 139% increase in yield over the course of one season, which is a huge advantage. In fact, increased yield is a very desirable quality for not only open field production, but for controlled environmental agriculture production as well. Here is a hydroponic tomato greenhouse. And so there's a lot of capital expenditure and cost to maintaining this controlled environment. And so it has become basically an industry standard to use grafted plants in these types of settings to increase yield and maximize profitability in a system with a high cost to operate. A lot of farmers have begun the use of high tunnels, which are a lower tech modified controlled environment, in which case they construct this sort of structure around a series of plants. The plants are planted in soil, so it's important to have disease resistance. But also this higher cost means that you want to increase your yield to maximize your profitability. So grafted plants for higher yield are also one of the main reasons that you would use this type of system. Another benefit is increasing fruit quality. So we have on the y-axis the kilogram firmness of watermelon and the x-axis weeks after harvest. So not only are the grafted plants having a higher fruit quality at harvest, but at 1 and 2 weeks after harvest, they maintain a higher quality than the non-grafted plants have produced, as the kilogram firmness of the watermelon for non-grafted declines at a faster rate, while grafted plants maintain a higher fruit quality. In North America, a lot of grafting is done in high capacity nurseries. Currently, about 58 million grafts are conducted in the entirety of North America. 63% of those are tomatoes, 24% are watermelon, and then cucumber, pepper, eggplant, and muskmelon make up the remaining percentages. In the year 2000, it's estimated that less than 100,000 plants were grafted per year in all of North America. So although grafting is widely adopted throughout the rest of the world, this is a relatively new agri-technology in this continent. But by 2012, this number has increased to 47 million grafts per year. However, less than 1% of those were conducted in the US. By the year 2019, this increased 23% to 58 million grafts per year in just 7 years, with the US now jumping to the number 3 spot with 13.5% of the total grafts in North America. Now, one of the limitations for grafting on this continent is that there are few high volume nurseries. In the USA specifically, there's only a handful of them that produce more than 1 million grafts a year. So if a producer is looking to purchase grafted plants, they have limited options, and a lot of those options are located far away. So a lot of producers choose to try and purchase plants from outside of the US as well. However, there are importation restrictions with plants being imported from Mexico. However, there are looser restrictions for Canadian grafts. And so Canada actually exports a lot of plants to the US annually. In fact, over 5.75 million tomatoes and about 2 million watermelon plants per year, which is 38% of all of the grafts conducted in Canada. Now, to get those plants from the nurseries to producers, they have to be shipped. In an optimal setting, they'll be shipped in trucks like this under climate-controlled conditions. However, that's not always the case. Up to 66% of these transportations are conducted without climate control, and the transportation time can take somewhere between 3 to 5 days, even under optimal scenarios. So while plants are typically loaded into these kind of trucks on shelves or packaged into boxes, a lot of them are shipped via box shipments like that of FedEx or UPS, or via air freight. Now, during that transportation, it's been highly reported that stress can occur to the plants in transition. One of the most highly reported stresses is chilling stress, which can reduce seedling quality and potentially affect development. These are plants from industry that were shipped for 3 days and experienced temperatures of 3 degrees Celsius. You can see significant necrotic tissue formation, browning, leaf spotting, some wilting and leaf curling. Now, these are the same plants transplanted into an open field in which we do see leaf curling and necrotic tissue formation. However, one of the most important characteristics for producers that judge the quality of their plants is their survival rate, which over 95% of these plants survived, but we don't know how this quality reduction impacted post-transplanting performance. So in the literature, watermelon is one of the most sensitive crops of those typically grafted. Of the cucurbits, it is the most sensitive and typically experience chilling stress between temperatures of 10 to 15 degrees Celsius. However, another study reported that you can store watermelon under low temperature conditions at 12 degrees C for up to 2 weeks without significant quality deterioration. So we'll return to this number later, as we're going to use that as a benchmark for part of our experiment. There are limited studies on the specific effects of chilling on vegetable crop seedlings. However, of those available, Cork as a default in 2001 published that as hours of 2 degrees C chilling increases from 0 to 81 hours, shoot and root dry weight measured immediately after the chilling stress is reduced, meaning that that plant is going to actually lose a lot of its mass and likely be inhibited once transplanted as it tries to grow vegetatively. Another issue is that these seedlings are also already expressing reproductive development. So here I have pictured 25-day-old seedlings, and these are watermelon buds that I have removed from the plant. And what we can see here are actually anthers from the male flower forming in the reproductive buds. Now, a lot of typical transportations are conducted when the seedlings are 4 to 6 weeks old, meaning that these structures are likely already present in the in the plant when they were transported. So this same study from the left did report that days to first female flower from transplant was actually delayed linearly as hours of 2 degrees C chilling increased to 81 hours, meaning that reproductive development was directly inhibited by chilling during that seedling stage. And in one of the 2 years studied, they did experience a delay in fruit set. Now, this could be a large issue for producers, as typically they want to have predictable harvest dates and then be able to conduct crop rotations in a timely fashion. And if they are harvesting later than predicted, that could be a big issue for them. So here's just an example of what we can expect with a transportation risk. So seeing as a lot of plants are shipped from Canada to the US for transplanting into the field, Southern Florida begins watermelon transplanting around mid-February every year, meaning that if you are shipping plants from Canada under conditions where there are not climate control on board during transportation, you can experience extremely low temperatures as the external environment will impact the internal environment of during shipping. So during that 3 to 5 day transportation, chilling stress can be experienced as plants move from a very, very cold region up north to the south, where they're able to be transplanted into more optimal condition. And so upon arrival and somewhere in southern Florida, they could, while they initially were shipped under a high quality condition, they could experience severe chilling damage and then potentially delays post-transplant. So we had three hypotheses going into these set of experiments. The first being that since grafting can often increase plant vigor, we think it might be able to also improve abiotic chilling stress tolerance. Secondly, longer durations and lower chilling temperatures would likely result in more seedling damage and longer delays post-transplant. And finally, we think that based on research that we can conduct, that an appropriate temperature for transportation can be found that maintains seedling quality and ensures that there aren't any developmental issues post-transplant. So our first experiment was 3 degrees Celsius chilling for grafted and non-grafted watermelon. We used SSX 8585. It's a diploid watermelon cultivar. And then we used strong TOSA interspecific hybrid squash rootstocks that are commonly used in industry. And we had 3 distinct stages of this experiment. The first being the seedling growth and the grafting stage, followed by chilling, and then followed by greenhouse growth. So this post-transplanting into a greenhouse was specifically selected for 2 different reasons. The first being it allowed us to conduct experiments year-round in a controlled environment. And the second being it would mitigate any possible stresses that might occur outside in an open field setting and allow us to specifically study the effects of chilling stress on the transplanting development. So here's just a timeline of how the experiment was conducted. In the first week, we germinated watermelon that were to be grafted. And in the second week, we germinated the rootstock that grew at a faster rate. We also germinated non-grafted watermelon 1 week later than the grafted ones, so that we could conduct grafting and healing over a week. Then we hardened the seedlings for an additional week before exposing them to chilling stress. And following that, transplanted them into the greenhouse to assess their vegetative and reproductive growth. The chilling treatments were conducted in growth chambers, and each grafted and non-grafted plants were randomly assigned to chilling containers and then assigned different treatments. So we conducted this chilling trial over a 72-hour period. That 72 hours was meant to mimic or simulate a actual transportation, typical time that you would take from the nursery to the producer. Our first treatment was 48 hours of 3 degrees Celsius chilling. And we also had 24, 12, and 6 hours of 3 degrees Celsius chilling. We aligned all of these treatments to end at the same time. And then on either side of this chilling challenge temperature, we had 12 degrees Celsius storage of the seedlings, which we know from literature to be a safe temperature for preserving seedling quality. And so we thought this would be a non-impactful temperature. And then we also had a control group with no chilling, no 3 degrees Celsius chilling, and just 72 hours of 12C storage. Post-transplanting, we transplanted into the greenhouse. We had 5 double rows. There were 100 total plants organized into 10 blocks, as each double row was bisected into a north and a south section, or a north and a south block. Each block had 5 grafted and 5 non-grafted plants, 1 of each from each chilling treatment, and there were a total of 5. The planting density was 3.25 plants per square meter, and watermelon is typically grown prostrate along the ground, and since it is a binding crop. So we did what a lot of large-scale hydroponic vegetable greenhouses do, and we trained the vines upward to horticultural twine supported by vine clips. And so they were grown essentially just towards the ceiling. To assess the health of the seedlings, we observed the health of all of the true leaves visually. And we analyzed true leaf damage and the difference in damage between before and after chilling. We did this by scoring them using the Horsfall-Barrett scale, which is commonly used in plant pathology, where each linear number, the linear scale, which each number corresponds to a % damage range that is a specific interval size, and then can be converted to an interval midpoint so that you can compare percent damage much more easily, which is a common transformation to analyze this data. Now, this was done in part to reduce any observer bias or error, as it's very difficult to tell whether a plant has 30 or 40% leaf area damage, but it is much easier to tell whether it falls into a 12 to 25 interval range or 25 to 50% damage interval range. Now, to give an example of what this scale looks like, this is a 1 or a 0% leaf area damage. There's healthy tissue and no obvious signs of deterioration. This is a 6. We have necrotic tissue formation somewhere between 25 to 50% of the leaf area. A 9 is about 87 to 94% of the leaf area damaged. And then a 12, in this case, is a dead plant where all of the leaf tissue area is damaged and there's no healthy tissue remaining. Now, just one other point is when we do this scoring, I'll be referring to this transformation, this delta midpoint is delta MP, which is the difference in damage from before and after chilling, in which case if there was 5% leaf area damage before chilling and 35% after chilling, then we could say that that 30% is directly, the 30% damage is directly a result of the chilling stress. After transplanting, we evaluated the plants vegetatively and reproductively. Vegetatively, looking at the primary vine length, the number of nodes on the plant, the average internodal length, and the leaf counts of the primary, secondary vine, and the whole plant. Reproductively, we looked at the days from transplant to first male and first female flower and thesis on each plant, as well as the node of the first male and female flower and where they developed. Just to return to the timeline for a moment, we did the injury scoring just before chilling and then 24 hours after chilling to assess the difference between these 2 points. And this 24 hours after was to allow the plants to visually present that damage a little bit better. And during the greenhouse growth, we measured vegetative measures weekly and reproductive measures daily. So into the results for experiment 1. Essentially in this experiment, we didn't see any trends with increased durations of chilling for either grafted or non-grafted plants. However, we did see a higher damage for grafted plants over non-grafted plants, likely due to the increased stress of healing just 2 weeks prior. Vegetatively, grafted plants did produce more leaves on the whole plant and had longer internodal lengths, so they were slightly more vegetative than non-grafted plants. And the chilling duration at 3 degrees Celsius actually didn't affect any vegetative measures, so vegetative growth post-transplant was unimpacted by the chilling stress. Looking at reproductive growth, we didn't find any effects of grafting or chilling on the number of days to floral development. Further, that indicates that we need more hours of chilling or lower temperatures to induce the kind of stress that we wanted to see that delays flower development. So from the first experiment, we can conclude that the variety we selected is not particularly sensitive to cold temperatures, that grafted seedlings are slightly more susceptible to chilling damage, as we saw with the seedling damage being increased, and that watermelon can generally tolerate up to 48 hours of 3 degrees Celsius without suffering long-term effects. So moving to the experiment 2, it's essentially the same experimental design as the first experiment, but with 1 degree Celsius chilling instead. We did make a couple modifications. One being the watermelon cultivar used was a triploid variety Tri-X313, which is commonly used in industry. We had 2 replications, one from, going from the winter into the spring and one in the summer. And then we had added a chlorophyll fluorescence measurement to analyze the health of the leaf tissues. And the chilling treatments were reorganized to have the chilling stress temperature, 1 degree C, occur first, followed by 12 degree C storage after that initial stress. And this was to avoid any potential acclimation to chilling temperatures that might occur by the 12 degrees Celsius chilling coming before the 1 degree Celsius chilling. So when we look at the results in the winter replication, we found that seedling damage did increase linearly for grafted and non-grafted plants as the duration of exposure to 1 degree Celsius increased to 48 hours. So this delta NP did increase, and grafted plants did show a more profound increase than non-grafted plants, again, likely due to the increased stress of healing just 2 weeks prior to the chilling. We also looked at FVFM, which is essentially a measure of the maximal possible light harvesting ability of the plant. So by shining a strong light on the leaf tissue and measuring how much light was actually fluoresced by the plant, meaning how much of it was actually utilized by the light harvesting complexes, we'll get a ratio basically saying what percentage of that light was actually used, and that indicates the leaf tissue health. So in this case, a 0.72 means that 72% of that light was being harvested. And in this case, that's about a healthy rating, as watermelon is typically between 0.72 and 0.78 in a healthy state. But we did find that this decreased linearly as the amount of chilling increased. And this was measurable directly after the chilling treatments were applied, meaning that this chilling stress had affected the plants, and we were seeing a reduced capacity of the plants to harvest light energy, which amounted to a total of a 10% decrease in the most severe treatment. Now, vegetatively, we didn't find any effects of the chilling stress on vegetative growth, similar to the previous experiment. However, grafting and replication did have effects. So we did see more vegetative growth in the summer replication, as there were higher temperatures and light intensities that were more optimal for watermelon. So we did see more growth in the summer. Grafting did produce longer vine lengths for grafted plants over non-grafted plants in both the first and second replications. Also, grafting also led the first flower nodes to be produced on nodes further along the primary vine, indicating that there was slightly more vegetative growth before the actual flower development was reached. And then in the summer, vegetative growth was, or reproductive growth was much faster, and the first flowers for grafted and non-grafted plants emerged on lower nodes on the primary vine. So when we look at the actual days from transplant to flower emergence for female flowers and the reproductive growth, we did find a difference between the two replications. In the first replication here, the female flowers were delayed for grafted and non-grafted plants, and there was a linear increase with the duration of chilling. At 48 hours of chilling, grafted plants had delayed female flower development of 3.2 days, while non-grafted plants were delayed 7.2 days. In the summer, however, there was no significant increase in the number of days to flower, and the plants did emerge sooner than, the flowers did emerge sooner than in the winter replication. Emergence of the male flowers followed a similar trend, where the grafted and non-grafted plants were delayed up to 5.3 and 6.2 days respectively in days to male flower with 48 hours of chilling, while in the summer, we didn't see a trend at all. And we have a couple theories as to why this was. The first being the growing conditions. So for each replication, during the seedling growth stage and the post-transplanting growth stage, we had a difference in air temperature throughout the greenhouse. In the second replication, we had higher daytime, nighttime, and daily maximum temperatures, as well as when we look at the solar daily light integral or light intensities experienced, we did have higher light intensities in the summer for both the seedling growth stage and the post-transplanting growth stage, meaning that this is more optimal condition for watermelon to grow and may have advanced growth quite a bit. What we wanted to look at was the chilling and its potential effect on reproductive development being different between these two replications. So on the left here, this is a cross section of a watermelon seedling in which we can see the apical meristem or the growing shoot tip being protruding up into the left here. And off to the right is the first flower bud forming on this plant. Now later on, this will develop into a flower bud like the one we saw earlier with anthers present in the actual bud. However, there's not any really good evidence about exactly how chilling affects reproductive development in different seedling stages. So this is, so reproductive development and chilling stress in the seedling stage is not well understood for cucurbits. However, when we look at other species like strawberry, which has 14 distinct flowering stages before actual flower development and emergence, 4 of those 14 stages are highly susceptible to chilling stress at or below 2 degrees Celsius. while the other 10 stages, when exposed to chilling stress, don't produce any reproductive delays for that plant. So a similar possibility may exist for watermelon, where certain stages are more susceptible than others. And we think that high temperatures and light intensities might be key to this. In the seedling growth stage, we think that higher temperatures and light might advance seedlings to a more chilling tolerant stage by advancing that reproductive growth more so than in plants grown under lower temperatures and light. And then post-transplant, regardless of exactly how much delay we would expect to see of the seedling growing conditions beforehand, there might also be that second factor that more optimal conditions post-transplant could increase the development rate of those plants and mitigate any possible chilling effects that we would have seen in the seedling stage. So our conclusions in this experiment were first that 1 degree Celsius chilling does cause visible seedling damage and decreased chlorophyll fluorescence, both in predictable patterns. However, neither of these effects had any influence on the vegetative growth, and vegetative growth was generally not affected by chilling. But chilling did increase the number of days to flower and thesis on the plants, with grafted plants delayed up to 5.3 days or up to 3.2 days with male and female flowers respectively, and non-grafted plants delayed 7.2 or 6.2 days with male and female flowers. So we followed this experiment up with a mixed chilling trial with the goal of examining how different durations and temperatures would affect these plants. So the hypothesis for this was that chilling stress might actually be more like a cumulative response, meaning that different durations and temperatures could be combined to create similar chilling stress that's measurable. So I'm going to draw in data from outside of this study. So this is actually a post-harvest basil storage study, in which case basil is stored at different temperatures after being harvested to try and maintain marketable quality for longer periods of time. In this case, 5 temperatures were used to store harvested basil, and then the basil was evaluated by using an injury score from 0 to 8 to assess marketable quality. At a level of 5 and above 5, the basil is no longer of marketable quality. So you want to maintain the basil below this line to ensure that it's sellable. Now we can see that this 5 is reached after different durations of different temperatures. And so we thought that maybe this cumulative chilling response could be modeled by an equation. So this is chilling degree hours, in which you're taking the sum of the difference of a safe temperature, which we call the base temperature, and the difference of the base temperature from the actual temperature experienced, times the amount of time that it's under that temperature duration. And by summing up all of those chilling degree hours, we're essentially able to create one value that is comparable between different durations and temperatures. So in this case, it appears that basil is stored safely at 10 degrees Celsius, so that we consider 10 degrees Celsius a safe temperature, and it accumulates 0 chilling degree hours per hour of storage at that temperature. However, when we look at 5 degrees Celsius, we would say that accumulates 5 chilling degree hours per hour. And then at 0 degrees Celsius, 10 chilling degree hours per hour. So for example, if you wanted to reach 50 total chilling degree hours, you could store basil at 9 degrees Celsius for 50 hours or at 8 degrees Celsius for 25 hours. And the idea is that this base temperature could be different for different quality parameters and for different, and might be also species specific for different storage temperatures. So we essentially took all this data and we transformed it into a CDH accumulation curve. So CDH with this base temperature of 10, so CDH 10 on the x-axis, and then the y-axis the same injury score. And we found that the points all fit nicely into a logarithmic curve like this, in which case we want to look at this 5 for marketable quality. We find that 5 is reached after 480 CDH10 is accumulated. And you could reach this same point of 5 with 53 hours of 1 degree Celsius storage of basil or 80 hours of 4 degrees Celsius storage of basil. In this case, we think that potentially watermelon seedlings that are chilled for different lengths of time and at different temperatures may be able to form a predictable response, somewhat like this. And perhaps that specific base temperature that's considered safe could be different for FVFM and seedling damage and the actual delays in flower development. So we organized this experiment slightly differently. We only used non-grafted watermelon, that Tri-X313, to mitigate any possible rootstock interactions that might occur during this chilling. And then we had 7 chilling treatments. We had one control, just about 12 degrees Celsius, for that total of 72-hour storage that simulated transportation, as we've done previously. And then we had 6 treatments between -0.4 and 1.2 degrees chilling for between 24 and 48 hours, with the remainder of the time stored at 12 degrees C. We then took the actual data of the temperature storage and then converted all of it to chilling degree hours using base temperatures between 3 and 15 degrees Celsius. By using these different chilling degree hour calculations, we're able to fit the data multiple times and discover where exactly is the best fit. And that best fit indicates that that is the appropriate base temperature to measure that quality parameter. So from the results, we found that seedling damage, FVFM, and female flower development were all affected by chilling. However, vegetative growth, the note of the first flower, and male flower development were unaffected. So when we look at seedling damage, we found that the best base temperature to analyze this data is 15 degrees Celsius. And while we've seen linear curves in a linear response in the past with seedling damage, as CDH15 increases, we see that there is after accumulation of 600 chilling degree hours, there's an exponential response, meaning that after a certain threshold is reached, the watermelon are producing a facultative response and are increasing in damage exponentially after this point. So they're tolerant of the chilling stress below 15 degrees Celsius for a certain amount of time. When we look at FVFM, we did find a linear decrease again. And the best base temperature to analyze this was at 3 degrees Celsius, suggesting that FVFM begins to be reduced at or below 3 degrees C. And FVFM decreased by 0.025 with every 50 CDH3 to a maximum of a 9% decrease in the most extreme chilling treatment. And when we look at female flower development, we found that the best base temperature was at 4 degrees Celsius. And this linear increase in days from transplant to first female flower was highly correlated with that 4 degrees C calculating temperature. And so female flowering was delayed approximately 1.3 days with every 50 CDH4 accumulated. Now, we think that this is as we found this is the best base temperature for predicting female flower development at 4 degrees C or below. However, we wanted to kind of check ourselves by looking at previous data that we'd collected. So this is the data from experiment 2 with up to 48 hours of 1 degree Celsius chilling on the x-axis here. And then the grafted and the non-grafted delays that we saw in the winter replication. Now, when we fit CDH4 on the x-axis and we fit our mixed chilling model prediction, we find that it falls neatly in between both the grafted and the non-grafted delays that we actually experienced. Now, we wanted to check this data and confirm these results outside of our own predictions as well, as this looks like a rather good predictive model to explain how flower development is impacted by chilling. So we're going to pull back to this data from Korkmaz and Default in 2001, which I'll refer to as KD, and they have 2 years of 2 degrees Celsius chilling of seedlings that are delaying the first female flower development as duration increases to up to 81 hours. So this is that previous graph where we see our grafted plants from the 1 degree C chilling trial and the non-grafted plants. And then we see the mixed chilling trial in the middle here. And then when we fit this cork mass and default data in 2 different years with 2 degrees Celsius chilling, we find that the CDH, when converting all of this duration to CDH, we find that in year 1 and 2, there's a really nice and close fit to the actual mixed chilling model that we created. So this indicates to us that 4 degrees Celsius is a temperature that begins to induce chilling stress, and that lower temperatures can use that chilling degree hour model to predictably evaluate how much days you expect to experience after transplanting, and not only transplanting into a greenhouse like we did, but also into an open field environment. So to bring this back to the hypotheses that I mentioned at the beginning, we found that grafting can improve chilling tolerance, as flowering is not as delayed for grafted plants as non-grafted plants are. We also found that longer durations of chilling and lower temperatures result in more seedling damage and longer delays, which we explored further with our chilling degree hour assessments. And then also that an appropriate temperature for transportation can be found, which I'll go into in a moment. So to conclude this, right, we found that watermelons suffer from seedling damage at or below 15 degrees Celsius, and that FVFM is reduced at or below 3 degrees Celsius. However, neither of these actually impacts the vegetative development of the plants after the chilling is induced, but instead flower development is directly delayed by temperatures at or below 4 degrees C, which means that when we're conducting long distance transportation of seedlings, we should target temperatures above 4 degrees Celsius to maintain post-transplanting seedling quality. And specifically, we should take measures to limit any accumulation of CDH4 during the actual transportation. And not only is this a good measure to maintain seedling quality, but also economically, that means that when shipping from one location to another, you only have to heat the trailer to at least above 5 degrees Celsius to maintain that high seedling quality, instead of heating up to typical transportation temperatures that are somewhere between 12 and 15 degrees C. So essentially, we want to make sure that we maintain seedling quality, and we can do that best by maintaining long distance transportation temperatures above 4 degrees Celsius. So I just wanted to acknowledge a few people. First of all, my funding source, the USDA, has been fantastic in supporting this project. Also, my committee members, Dr. Kubota, Dr. Sheerans, and Dr. Kleinhens have been absolutely wonderful in helping me get through this. I also want to thank the Department of Horticulture and Crop Science. Dr. David Barker helped a lot with statistics. Dan Gillespie was very helpful when he was here helping with my project. Sakata Seed America and Syngenta both supplied seeds to us, which we're very grateful. And to the Kubota Lab in general, Mark, Gio, Madeline, Mary, Jason, Broderick, and Kazuya, thank you so much. I couldn't have done this project without your guys' help. So with that, thank you all so much, and I'm happy to take any questions.